Hello everybody, welcome to Dr. Sam's Anatomy classes. I'm Dr. Azmi Mohsin, Professor of Anatomy, and in this lecture I'll be teaching you about the histology of cerebrum, right? Okay, so before starting teaching you about the histology of cerebrum, we should have some idea about the cerebral cortex. Okay, so here you're seeing, this is the supralateral surface. So on the inframedial surface of this temporal lobe, you find there is this innermost gyrus, the parahippocampal gyrus. The anterior portion of that on the medial surface was there was incus, right, and amygdala even medial to that, right, hippocampus. So that hippocampal formation and the piriform area, and the joining areas together, there was secondary cortex, the entorhinal area, all this together, like primary and secondary olfactory cortex together. This portion, like I have drawn it in dotted lines, means it's not on the superficial, rather on the deeper surface. So remember that, and this is called rhinencephalon. Got it? And this rhinencephalon, remember, is a part of allocortex. This is allocortex. Allocortex is also called archipallium. So you've seen that this is the most primitive cortex, cerebral cortex, and it's right from the lower animals, even present in human beings. So it's primitive. Then remember that this portion of the cerebral cortex has only 3 layers, and it is a thin cortex. Got it? Now the rest of the cerebral cortex you're seeing, this is all called neocortex. Neocortex is also called isocortex. This is comparatively thicker cortex, and it has 6 layers. Okay, so this is undoubtedly, when we talk about cerebral cortex, it's presumed that we are talking is about the neocortex. And this neocortex, again, remember that it has 6 layers, I've told you, but there are different types of, let's write it down so it becomes easy. So this has various types of cells. You have neurons in this. You'll have Neuroglia, like the connective tissue of the CNS. Then you have blood vessels, right? So all this you're going to find in the cerebral cortex. But the difference is that there is unequal distribution of these neurons. Some are sensory, some are motor in function, but that is not equally distributed. So let's compare it with the cerebellum, right? So we are going to compare cerebrum with the cerebellum. Okay, so look here now, this is cerebrum, and pons, this is a brain stem, right? And this you're seeing is the cerebellum, right? Here you have vermis, and this from vermis there is this is all sulci radiating out like this. So the cerebellar cortex is, remember that it is called as homotypic cortex. Homotypic cortex means it is 3 layers, like remember that it is 3 layers, and it has equal thickness throughout the surface of the cerebellum, and the cells are also homogeneously distributed. Unlike this portion here we're talking about is the, we're just leaving it out, this portion, but this much portion, as I taught you, that it's neocortex. So this comparatively, comparing it with the cerebellar cortex, this is heterotypic cortex. This is heterotypic cortex. Now, why heterotypic? Because it has, the thickness of this cerebral cortex is unequal throughout its length. This thickness of cortex is around 1.5 millimeters to 4 millimeters. So the thickness of this cerebral cortex is different in different areas. If you remember, the roof of the fourth ventricle that was being formed by superior medullary velum and inferior medullary velum. Superior medullary velum although had a thin sheet of gray matter, but this inferior medullary velum was devoid of these neurons. So some places there is very less of cerebral cortex. So that's why, because of one reason is like, having unequal thickness, the cerebral cortex, and another thing is the cells here. I told you that this is having 6 layers, and in 6 layers the cells are also not evenly distributed throughout. There are some areas where you have more of granulocytes, more of sensory cells, where some places you have more of pyramidal cells or even differently specialized cells. So because of this unequal distribution of the cellularity and unequal thickness, it is called heterotypic cortex. Okay, so just get familiar with these different little words which I am telling. And before starting the histology, let's make you remind about the cells. If you remember, like in humans, like if you go to the first lecture of my neuroanatomy unit, there I have taught you about the the cells, the different types of neurons, and there I told you that in humans there are around total of around how many cells, if somebody asks, neurons in a body, that's actually 100 to 200 billion. We have 100 to 200 billion neurons in human body. Okay. Now, if it's asked like how many, because this you have to have a comparative idea. So, in the cerebral cortex, we have only 14 to 16 billion neurons in the cerebral cortex. Again, always you have to have some acceptability in this range, because there are so many authors. It's all people, what we teach is actually from the different books and the research publications. Some books might have written it as 100 billion, some as 200 billion. So that's why I'm writing the range. So you have 100 to 200 billion neurons in the human body, out of which in the cerebral cortex we have 14 to 16 billion neurons. Okay. So this you have to remember, so that okay, the cell size I'll tell you when I'll be teaching you about the histological slides. Okay, now I'll be teaching you about the histology of cerebral cortex. Okay. So let's draw the cerebral cortex. Cerebral cortex, let's presume that it's the top layer, right? And this one is the lowest layer. Okay, so it's a prototype diagram. Keep imagining what I'm telling. So, and let's divide it into 6 layers. 1, 2, 3, 4. Hold on. Sorry. Okay, so let's draw this cerebellar cortex. Cerebellar cortex, cerebral cortex, right? So this is the outer layer, and then you have is the first layer this much, then the second layer, third layer, fourth layer, fifth layer, and the sixth layer. Remember that these are all imaginary subdivisions, because in in real, when you see the histological slides, you won't find these lines separating, not a very clear cut demarcation in all the 6 layers. So let's even mention it so that you remember the numbering. So this one is, let's say, number 1, number 2, number 3, number 4, number 5, and number 6. These are the layers of cerebral cortex. Then you remember that on the outer surface, I told you, was a thin layer of Pia matter covering, and that was also having 2 layers, right? The Epipia, the Piaglia. The Epipia was running above. Piaglia was, they had blood vessels surrounding here. These are the blood vessels, and there was Epipia running with this like this, covering them, and then there was Piaglia, which was like so much closely applied to the cerebral cortex. Okay, so now the first layer, that's the outer layer. Let's talk about the outermost layer, that's layer number 1. This layer, this layer is having parallelly running nerve fibers towards the periphery. Parallelly, longitudinally or horizontally running, and parallel to the surface of the cerebral cortex. These are on the superficial most surface of the first layer, and this surface also has some peculiar neurons. And let's why not write down the name first. This layer I'm writing in short because there's less of space. This layer is called molecular layer. Molecular layer, or the plexiform layer. Plexiform layer. It's all in short. I'll describe them in detail later on. Just remember that writing it in short, less of space is there. So this is plexiform layer. Now about importance about this, this is more of nerve fibers and less of cells. And the cells you find here is sometimes you find this peculiar type of cells present, let's see, and these are like spindle shaped cells, fusiform cells present here. And because these cells you're seeing, they have like horizontal projections. These are horizontal placed dendrites, right? So they have the horizontally placed dendrites, and because of the horizontal presentation, I think I should use a little more lighter color so that you can see it better. So this you're seeing is horizontally placed cells and their horizontal dendrites. Because of the horizontal placement, and they have a peculiar role in this layer. So let's describe these cells. So in the first layer we're writing here, it's describing here is the molecular layer. Molecular layer, the outermost layer, also called as plexiform layer, right? And these cells have nerve fibers, I have already told you. Then you have special cells here, and these cells, specialized cells, these cells are called horizontally placed, right? So these are called horizontal cells of Cajal. These are called horizontal cells of Cajal, right? Otherwise, they have a different name also, and that is called Cajal Retzius. Cajal Retzius cells or CR cells. CR cells. These are also called as CR cells. CR, okay, these are, I can write it as CR cells. Now a little description about these cells. What are CR cells? Okay, so let me tell you that these are cells, actually the horizontal cells of Cajal or the CR cells. They are having embryological significance. The CNS develops a neural tube that has like inner cell layer, the parenchyma that's formed from there. They have mantle cell layer, the marginal outer cell layer. So these cells were like it's the cell embryologically they are derived from the marginal cell zone. They are present in the marginal cell zone in an embryo, right? And it has a role in the development of the CNS. So in the fetal life, the role is to help the neurons. They help the neurons in migration. It helps in migration and repositioning of neurons by cell to cell interactions. Okay, so that means if you remember, there are a lot of cells which have like migrated to get cells arranged in their definitive positions. And that position, if you remember the facial nerve, how it was winding around abducent nerve, that forming facial colliculus. So, that migration of the cell to different regions, like the basal gang, the basal nuclei, and they're interpressed between the white matter. So how these neurons, they get migrated to different regions, they have a role. Now these cells, horizontal cells of Cajal, help in migration of this. And now actually how does it actually helping it? By this cell to cell interaction, and that is by this peculiar substance that is secrete, and that the secrete is a glycoprotein. They secrete a glycoprotein, and that is called reelin, REELIN. Remember, this is the glycoprotein reelin. Remember, this reelin is important, and this reelin is a glycoprotein which is secreted by this, which has like cell to cell interaction, and because of it helps to migrate the neurons to different places. And now because this reelin, because of the research work going on, and they have found that this reelin has a role in underlying different degenerative diseases. Like they have a role, this reelin has a role in Alzheimer's disease, and it also has a role in autism and temporal lobe epilepsy. So these are different degenerative diseases where it has been researched and found out that reelin as one of the glycoprotein which is also responsible in these different diseases. So that was about the horizontal cells of Cajal. Remember this, the importance about these cells. Remember they are present in the molecular layer of the cerebral cortex. Now because we have come around talking about these Cajal cells, why not we discuss about another cells? There are also Cajal cells present in the human body, because maybe I get or do not get time to revise it again. So let's describe about the other different type of Cajal cells, and those cells are called interstitial cells of Cajal. I believe you must have heard, those who are preparing for PG exams. These are interstitial cells of Cajal. Because of the similar name, I am just describing it here. These are interstitial cells of Cajal, right? So, interstitial cells of Cajal, where do you find it? Any idea? You remember the layers of the intestine. Now, these cells are present in the intestine. So, in the, there are the 3 layers in this intestine. The 3 layers of the intestine. The inner layer was the mucosal layer. Then you had the outer layer as the submucosa. You had the submucosa there, surrounding to that was submucosa. And what was the muscle layer? Can anybody guess? There are 2 muscle layers. One was the inner circular muscle layer. These are inner circular muscle fibers, right? And then you have the outer, because this is a transverse cut section, so your outermost will be like longitudinal muscle fibers. So these are outer longitudinal muscle fibers. And the last one will be the serosa in some cases, or in some cases it will be tunica adventitia. The peritoneum covered structures have the outer layer serosa, which are retroperitoneal. You will have tunica adventitia as the 4th layer. Now the important thing is, which we are going to discuss, is about these cells. Now these cells is again like fusiform cells. Remember, these are the interstitial cells of Cajal. If they asked, where do you find the interstitial cells of Cajal? The answer will be, remember, you find them in the circular muscle coat of intestine. This is the site where you find interstitial cells of Cajal. Okay. Then what is the role of this? And if you remember, those cells, like the horizontal cells of Cajal, they were like neuronal in origin, right, from the marginal zone of this neural tube. And these cells, remember, are mesenchymal. These cells are mesenchymal in origin, the horizontal cells of Cajal. Now, what the role of these cells is actually, they help in communication, like, everything, like, the nerve supply is there by autonomic nervous system. So, it communicates the autonomic nervous system with the smooth muscle cells of this, smooth muscle cells of this intestine. So, it's sort of communication between the ANS and the smooth muscles. So, now again, if they have like any degeneration of any these cells, the interstitial cells, they will be underlying diseases. Now, can you expect what will be the diseases if there is like degeneration of these interstitial cells of Cajal? What can be the diseases? Like, there will be paralysis, gastroparesis, can be there, dysrhythmias can be there. Dysrhythmias can be there. Then there can be like, slow intestinal transit. Because of the degeneration of these cells, especially in diabetes mellitus patients, right? Diabetic gastroenteropathy. These are certain conditions where you find the underlying cause might be the degeneration of the interstitial cells of Cajal. Okay. So this is what about the two cells I have told you, and about these horizontal cells of Cajal. One important thing that was left out here is, remember, the not here actually, you should write it. The neurotransmitter secreted here is, I should use the same pen so you can remember, this neurotransmitter secreted is which neurotransmitter these horizontal cells of Cajal secrete. Remember that it is glutamate. This is important. These are few things which I am underlying because they might become a question sometime in the form an MCQ. So this is glutamate. Okay. Okay, so that was about the cells. Now we are back to this. Yes, we were talking about this, right? The cerebral cortex. Okay, now the first layer is done. Now we will be talking about the second layer. And the second layer, is called the outer granular layer. Outer granular layer. So as the name suggests that these are granular cells, right? So if they are granular cells, they will have granulocytes here, right? But small little cells, these are granulocytes, and actually they are stellate shaped cells, but because being so small, they are stellate cells. They are also called stellate cells, but because they are so small that they appear spheroidal. Got it? And because they're stellate cells, they have their radiating out, dendrites like this, right? So they have radiating out dendrites, and remember that the dendrites, may reach above also to the layers down below also, and they may communicate to the adjacent neurons. So that was about this layer, the abundance of these layer. And don't forget that, as I told you before, that the cerebral cortex is made up of mainly these two types of cells, granulocytes and pyramidal cells, and they are present in all the layers, right? Except I'll tell you that pyramidal cells will not be present in the sixth layer. Otherwise, it's present here also. So, and these pyramidal cells, they appear as triangular cells, small triangular cells. But because it's a granular layer, so you'll find less of pyramidal cells. But remember, like, exam time, MCQs time, so don't get confused. Pyramidal cells will be found in all the layers except the sixth layer. That's how you remember. Okay. So, and pyramidal cells, they appear triangular with the apex towards the periphery and the base downwards. And they have, their dendrites arising from the three corners, and they descend down also. So this is how, the axonal process from the base descend downwards. That was about, and don't forget, less of space is there. I need not write every time. Remember that the glial cells, like, they will be present throughout the different layers of the cerebral cortex everywhere. So that was about these, granulocytes. And why not, okay, leave this, I'll write separately. Yes, so now let me tell you that the next layer is outer pyramidal layer. Okay, why to leave this layer? Let's describe a little bit about this. Now the second layer, as I told you, was outer granular layer. Outer granular layer has, I'm drawing it a zoom type diagram. So these are the different types of cells you will find there. They are actually stellate cells, but because they're so small, like, so they are actually looking, they appear as spheroidal, and they have like radiating out dendrites. And remember, because the dendrites are, spread out radially in different directions, and they're small dendrites, and they are confined to the gray matter. So can anybody guess what will be, the type of these neurons? Will they be Golgi type 1 or Golgi type 2? So they will be Golgi type 2. Remember, again going back to my first lecture, the types of neurons that I told you, that Golgi type 1 are those, because multipolar neurons, both of them, Golgi type 1 and 2. The one was those which had like a long axon, like in a long process, this dendrites were small, and the one you have a one long axon, that was reaching into the white matter. So that was that was part for the multipolar Golgi type 1 neurons, the axons forming a part of white matter, or the part forming the part of peripheral nervous system. But Golgi type 2, they are, the axons and dendrites nearly the same, and they are restricted only to the gray matter. They do not extend beyond the gray matter. So they are Golgi type 2. Remember this. Okay. Okay. Now let's move to the third layer, and here I'm going to talk about is the outer pyramidal layer. As I told you, now in this, the name is pyramidal layer, so you will have more of pyramidal cells, right? And pyramidal cells are triangular in shape, right? And they have a, why not describe all it here? The third layer is outer pyramidal layer. Outer pyramidal layer is made up of mainly of pyramidal cells, and these pyramidal cells, I told you, are triangular in shape. They have their dendrites which might reach to the layers above, right? And they spiculated. When you talk about the spiculated dendrites, so that means they are branching out, and they, from the two corners, they have their dendrites moving out horizontally, but the base will have a long axon. Now this axon will reach down to cross different layers, and this might even reach down to the white matter. So this is how a pyramidal cell is. And while we have written this, remember once again now, how many neurons I told you were there in cerebral cortex? Remember, they were 14 to 16 billion, right? Okay, now talking about the size, these, these pyramidal cells, they are the large cells, but they have a good range, 10 microns to 120 microns. That's the size, the longitudinal size here. No, not the longitudinal, sorry. Remember, what I'm saying is, when I'm saying is about the size, we are actually referring to this length. This is the size. This is the size. What I'm telling is the cells, or the size of the pyramidal cells is 10 to 120 microns in size. And because I left this out, why not know the size of these cells? Okay, so these are the cells. Here you see is the size of granulocyte. Now the granulocyte, of course, they will also have a size. They range like 5 to 8 microns in size. So they are comparatively much smaller. Even just see the smallest extent of this pyramidal cells is 10 microns. So even the smallest pyramidal cell is bigger than the granulocytes. Okay, and remember, granulocytes are receiving the afferents, while these are the ones which are giving out the efferents. So they are like the sensory neurons, and these are the motor neurons, outer pyramidal cells. Okay, so there you've done, and don't forget that again that you have granulocytes present everywhere. Don't get confused that these are only restricted to the layers. And I am not mentioning every time that glial cells will be present throughout the length. So these are the, they will have horizontal communications also. This was all about the pyramidal cell layer. Then now another important layer, and we're using the same color, so you have this idea. Now there is this inner granular layer. Now this is inner granular layer. Now inner granular layer, you will have again the granulocytes or the stellate cells. Now you can better call it as stellate cells because the size has become a little more bigger, and because bigger in size, so you can even appreciate their stellate shape. And again they have these dendrites giving out radially. This is how, and this is, now I am talking, is the third layer. Okay, first let's cover a bit here. The important thing what I am telling is that this layer has, on the inner side, you have a band of parallelly running band of white fibers. This appears as a white band, and that's because of the band thing, it appears, it's a striate appearance, and this band is called external band of Billarger, right? So let's discuss about this here separately. Now it's the layer we're discussing is the, yes, it is the fourth layer, and this one is inner granular layer. Inner granular layer, again the cells will be stellate and they will have like radiating out dendrites. Remember that these dendrites, why they are called spiculated? They call it spiculated because they have a ramifying dendrites, they branching dendrites like this. So they are spiculated dendrites. And the granular, why the granular cells? Because they have a granulated appearance, right? So that's the inner these are the cells which form the inner granular layer. And these granules are, these dendrites is ramify with the horizontally with the same layer cells of the upper layer cells of the layer below. And they might even reach, afferents coming from the different areas, the association and commissural fibers. Okay, so yes, the band of Billarger, I am to tell you. Now, as I told you, the inner portion or the deeper portion of this fourth layer has this band of fibers, the horizontal band of fibers, right? And this band of fibers seen in this layer is called the external band of Billarger. So these are the axons, like, they are coming from different associative areas, and they are distributed like this, and this can easily be like, seen sometimes with the naked eyes. Remember in the stria of Guinnari, that was the calcarine sulcus, there you finding the bands. So these are actually the bands in the cerebral cortex, and because they present comparatively, there is another band on the fifth layer. So that's why, because this one is an outer layer and this called is external band of Billarger. Okay, now we will talk about the fifth layer. The fifth layer is again using the same color. Now this layer has the biggest neurons in the human body. Important point is the biggest neurons in the human bodies. So they have like, their dendrites reaching above also, and their dendrites, ramifying here horizontally from the two angles, and their, axons, they descend down. And the axons which they descend down, they are I am drawn this arrow so that you remember that these are the efferents, like, and these fibers descending down from these large pyramidal cells, this will form the projection fibers. And so this is internal pyramidal layer or inner pyramidal layer. So let's describe this here in detail. This is the fifth layer now, and this is inner pyramidal layer. And this inner pyramidal layer, important is that these have the largest pyramidal cells. So these have dendrites reaching to the layers above. They have horizontal dendrites from the two corners of these triangular cells, pyramidal cells, and there will be a long axon arising from the base of these triangular cells. And these axons, they will form the projection fibers. And cortico-spinal, cortico-nuclear, corticobulbar, all these are projection tracts, descending tracts. So these fibers, they will descend down even to the basal ganglia and to the cells of the nuclei of the brainstem, then to the anterior horn of the spinal cords, right, and they decussate at this upper medulla and the lower medulla to reach to the contralateral side. So these are the and they will pass to the internal capsule, like, so they're really long neurons, and these are the projection neurons. Of course, they will all be myelinated by the oligodendrocytes. And now the important thing is that these cells have a special name because these are the largest cells, and remember their range in this layer, remember the fifth layer, because they are pyramidal cells of different sizes, 10 to 120 microns, but these are the largest ones, so their range is 100 and 120 microns. This is how much large cells they are, and they have the special name called Betz cells. These are the Betz cells. Okay, so now if asked which is the largest cell in the human body, I taught you this again in the first lecture of introduction of neuroanatomy. Again, you can go and go through this and revise. I discussed about this, which is the largest cell in the human body. Again, if it's asked, your answer will be the ovum, the secondary oocyte. The secondary oocyte that is liberated from the ovary, and it is that which is meant for fertilization. Got it? So you can here only again write it down so that it doesn't confuse you later on, that the largest cell of human body, if it's asked, the answer will be oocyte, or the secondary oocyte is the better word, or even sometimes they write this as ovum. Ovum can be the answer. That is actually that's liberated from the ovary and it's ready to be fertilized. So that's, and the size here, this ovum, this also has a varying size because you're talking as humans. This is around 120 plus in size, 120 microns. Greater than, a little you can say is greater, but the why not Betz cells call as the largest cells? The reason is they have a range, but this doesn't has this is one single cell at every month being secreted, but this has a lot of majority of cells, so they have a good range. So your answer will be again the largest cell of human body is oocyte, secondary oocyte. So that was about these projection fibers. And don't forget, I told you that on the inner surface, again, that the inner surface of the fifth layer also has this horizontal band of fibers running like this, and this is called inner band of Billarger. This again is a horizontal band running in the fifth layer. So you have two bands of Billarger if in case it's asked. So remember, external band and internal band. This also, internal band of Billarger will be in the deeper portion of this fifth layer, and this is internal band of Billarger, right? So this is internal band of Billarger. Internal band of Billarger. Okay, and so that you don't get confused, you always learn this habit of drawing stellate cells also in between, because these are also present in entire length in all the layers. And now the innermost layer. The innermost layer is having a lot of peculiarities. Let's use this special color. This is called Pleomorphic layer or Polymorphic layer or Fusiform layer. We have different names for this. And one more thing I just skipped, this is important. If it's asked what is the ganglionic layer of the cerebral cortex, remember this layer is also called ganglionic layer. Ganglionic layer. So why not write it here? Okay, this inner pyramidal layer is also called, inner pyramidal layer is also called as ganglionic layer. So in case if you get confused, remember ganglionic layer in retina also, if I remember, retinol, they were also counted from outside to inside. Let me tell you that whatever layered structure, if you have asked in histology, you have layers like in cerebrum, cerebellum, retina, the numbering always is from outside to inside or from periphery to deep. So in retina, the ninth layer, like remember the ninth layer was the nerve fiber layer or the retinal nerve fiber, but the eighth one was the ganglionic cell layer. So here also you have the ganglionic cell, because there the nerve fibers of the ganglionic cell layer in the retina, they were forming the second order neurons and they were actually forming the optic nerve. So here also in cerebral cortex, these ganglionic cells will form the axons of the corticospinal tract. And one more thing, if they ask what are these, they're different name, you can ask, they can mention. So don't forget that these axons of the large pyramidal Betz cells, they are also called as upper motor neuron. These are also called as upper motor neurons, because these will project on the anterior horn of the spinal cord or the cranial nerve nuclei. From there the efferents will emerge, the lower motor neurons will move out in the periphery to form the spinal nerves or the cranial nerves. So these are upper motor neurons in the axons of the Betz cells. Upper motor neurons. Now about this polymorphic, pleomorphic or fusiform cells. Okay, so these cells, as I was using green color, so let me tell you this special thing about is that these cells are fusiform, but they are vertically placed cells. Fusiform with vertically placed cells, and they will have their dendrites arising from the two ends of this fusiform cell. Now, another important thing is that their axons will arise from the center of this cell. That is the speciality of this fusiform cells. They have their axons arising from the center of the cell, and at the two ends they have the dendrites and they are fusiform. Another very important cells you find here, what color should I use now? I think red. Let's start with the red. Now another cells, now these cells are multipolar neurons. These are multipolar neurons, right? And these cells, about the peculiarity of these cells is that they are inverted neurons. These are inverted neurons. Their axons runs towards the periphery. They have the dendrites here in this pleomorphic layer. So these cells which have inverted neurons, they reach to the layers above. So important about this is, and let me tell you that these cells are Martinotti cells, right? Apart from this, because as the name suggests is pleomorphic, a polymorphic cells, don't forget to draw the granulocytes here also. Granulocytes, the stellate cells also, right? And I have not drawn anywhere the glial cells. Remember, the glial cells will be present in all the 6 layers. Okay, so let's describe this 6th layer. The 6th layer is pleomorphic layer or polymorphic layer or fusiform layer, right? And why fusiform? Fusiform, I told you, it has these special cells which are vertically placed fusiform cells in this layer. If this is the 6th layer, right? So they have, these are vertically placed and their dendrites will be projecting out from the two ends, and the important thing is their axons will descend down from the base or the center of the cells. These are the axons. This is how you find is this cell, and this cell is called fusiform cell. Right? One special cell is this in the plexiform layer, and the other cell, yes, so the other cell, as I told you, is, and again, hold on, so that you don't get confused, again solving your MCQs, Martinotti cells are present in all the layers. Simple, these are sometimes distractors, so but because I'm just telling you that you get solve your MCQs. So it's not peculiar, but because again you will this is a plexiform layer, you predominantly find these Martinotti cells here in the 6th layer, but it doesn't mean they are not present anywhere else. So as I had described about let me draw it first. So as I told you, the importance about this is they are the inverted neurons. They have the axons reaching to the layers above, and the dendrites will be present here. So these are inverted neurons, and these are, let me tell you that this word is an Italian word. So your pronunciation should be in Italian accent. So this is Martinotti cells. That's how you pronounce in Italian accent, Martinotti cells. Okay, and these are inverted neurons, and so that you don't get confused, it's present in all the layers, right? Okay, but fusiforms are peculiar of this layer, the sixth layer. And because here I am writing about these presence, so these fusiform cells are peculiar present in sixth layer. These are present in the sixth layer, while pyramidal cells, don't get confused, so I am writing it again, that pyramidal cells are present in all the layers except the sixth layer. These are sometimes as options given in your MCQs, and why leave the granulocytes in case if it's asked, like these are also called granulocytes, or also called as stellate cells. These are present in all the layers. These are present in all the layers. And why not? Because sometimes people take screenshots, so not write the name down here. This is the pyramidal cells. Okay, so the four major types of cells I have described there, so that you get help in your MCQ solving. Now, after going through all these layers you have seen here, now the afferents, I think I should bring out some new colors. Yes, white will be better. So now remember, what about the afferents? And I think I should use some bigger writing. So this is how you are seeing is the axons reaching above. These are the arrow I am drawing is here, that the afferents ascend up like this to the layers above and relay there. The arrow I am trying to write, right? And they will reach to these granulocytes. Hold on. As of course you can draw it reaching to any layers, but because there are granulocytes in this layer, so you can draw it like this, that they reach these arrows. So these are afferents reaching down to that granulocyte layer. But again I repeat that granulocytes are present in all the layers, right? And efferents are those which are descending down from the pyramidal cells, and you can find pyramidals in all the layers, but the efferents will be arising from the pyramidal cells. Okay, so that was about the neuronal tissue of the gray matter. And one more important thing apart from the glial tissue you will find is the blood vessels. In the slide, histological slide, you will also find lots of blood capillaries. Oh, sorry, this is something I shouldn't use. Yes, so this gray matter has a rich amount of perfusion from the blood vessels. So these cortical branches given out from the cerebral arteries, these capillaries, they descend down and along with the pia mater, forming perivascular sheaths, and they will form capillaries. Around these capillaries you find is blood capillaries. These blood capillaries you will find it is like this. A blood capillary may be like somewhat like this, and you will have a neuron on the side, the endothelial cells. This you can draw it like this. blood capillaries with the simple squamous epithelium. Sometimes two cells, the epithelial simple squamous cells, they make the endothelial lining of the capillaries. This is how you will find in every layer the blood capillaries. Okay. And now about the, yes, this was the 6th layer, and now about the neuroglia. Neuroglia present in the gray matter. One thing will be oligodendrocytes. Oligodendrocytes, they form myelin around those neurons, because don't forget that there are axons also in the gray matter. I have drawn so many horizontally running bands, external internal band, then it was horizontally running fibers in the outermost layer, the plexiform, and they are vertically running axons like in between the different layers. So there are axons and they will be myelinated by the oligodendrocytes, and the other cells they will find here will be these type of cells. They have protoplasmic extensions, and these cells are called offshots. These cells are called protoplasmic astrocytes. These are protoplasmic astrocytes. And third layer will be amoeboidal type of cells, and these cells are these oligo and protoplasmic and astrocytes, they were neuronal in origin, right? But this one is microglial cells. And these are phagocytic. One thing, these are mesodermal, and these are phagocytic cells. While protoplasmic astrocyte, what did they do? They form blood-brain barrier with the they surround those blood vessels. Then endothelial, I have drawn so many capillaries in all the layers, so they surround these capillaries to form the blood-brain barrier, the protoplasmic astrocytes, and also they are nutritive in function. Okay, so that was about the glial tissue in the cerebral cortex. And before I complete, there's less space here. Anyways, because you must have copied everything, taken a screenshot or whatever. I think because there's less of space, I need to rub this so as to draw a little bit of the white matter also. So this was the first layer, the plexiform or molecular layer. Now this leaves some space for the white matter. So as I told you that there were fibers running like this, 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 there were fibers running like this. They have interwoven meshwork of fibers, and this forms the basis of white matter. It doesn't mean there's no gray matter. The gray matter also sometimes, remember the migration of neurons. So sometimes the gray matter nuclei present the white matter, but that's a different thing. We are just focusing on the white matter. So this you see is a bundle of meshwork of bands of neurons, and that forms the white matter. This is the white matter. And don't forget again that this will also have blood capillaries. You will also find blood capillaries down there, right? And they might be, the branches from, the capillaries derived from the central arteries. And need not to say, I have not drawn less of spaces there. There will be neuroglial cells here also in the white matter. A short description about the white matter is also the last portion here. That is about, This I'm talking is about the white matter. White matter is, axons forming tracts, and they may be association fibers, they may be commissural fibers, they may be projection fibers. Like you must have been taught about how these association connects different areas of the same cerebral hemisphere. Commissural connects two different commissural hemispheres, crossing fibers from one to right to left, and these are the ones which descend down to the nuclei below, like in the brain stem to the spinal cord and in the extra nuclei of the extrapyramidal tracts. Okay, so those are the projection, the vertically aligned fibers. Now about the glial cells in this tissue. Glial cells of white matter. Again, here one cells will be oligodendrocytes. Oligodendrocytes will be there. They will be myelinating adjacent neurons, but not a good thickening. And compared to the Schwann cells, they are like multiple layer forming, myelin and neural lemma. Here, a single Schwann cell was encircling different peripheral axons completely, and here one oligodendrocytes encircles and myelinates adjacent, axons, few axons. So this one forms a thin layer, anyways. Then the other thing is, yes, the important one is here, that these astrocytes, I was to tell you that the astrocytes, if they ask, now there are two types of astrocytes in the glial tissue. Now these are called as fibrous astrocytes, right? The fibrous astrocytes, again, they will form blood brain barrier, but in the white matter. And the third one will be microglial cells. Microglial cells, again, these are mesodermal in origin, and they are phagocytic. Okay. So that was about the cerebral cortex. Okay, what is remaining here? Anything, if it remains, I don't remember everything I have taught you. Okay, so that was all about the histology of cerebrum. Hope you've understood it, and if you have any difficulty, you can write down in the comment section and ask queries, whatever you have. Okay, and if you are uncertain about something, you can rewind this video to check it from before, backwards, before, again, rewind them, and you are free to ask your queries. Okay, thank you so much. Okay. Okay, before winding it up, I thought of including the slides also in this, so that you don't have to search for the slide identification, because that was what I was taught is all the theory and text matter, but of course you need to know to identify the slides also. So I think I should include the slides. So at same place in the same lecture, you can find the slides. It will be easy for you to find the slide in the same diagram. So here saying is the histology of cerebrum, right? So histology of cerebrum, because everything I have taught in detail, because no doubt this will be a long lecture, but in your future, for any time you can revise with this. You don't have to search of the slides anywhere else. So I've included it in this. Now look here about the histology of cerebrum, how you will find the slides, right? So here you seeing is histology of cerebrum. As I told you that the layers, the 6 layers are not very easily demarcated. You cannot differentiate very easily. But what you'll have to find actually, some things you'll have to need, before identifying the features. One thing is like an outer surface. I told you there will be some collagen, the pia matter, the connective tissue type of thing you will find here. This like connective tissue, you can identify that this will be a pia matter, right? Then the molecular layer, of course, it's not fully fibrous. That's why it's called plexiform layer as well. And those horizontal cells of Cajal, you will not be able to find very much, because I told you the Cajal cells. Hold on, I didn't write. I think that the important thing about Cajal cells. Have I written? Yes, the horizontal cells of Cajal. Apart from this, one thing more important was left, that these cells are, they die out. As I told you, the purpose of these cells has a role in, they undergo apoptosis. There's less of space actually. Undergo apoptosis. Yes, the important thing is that these cells, as the purpose of these cells was to migrate the neurons to their destination during the embryological period by cell-to-cell interaction with the reelin. But after they have done their job in the postnatal life, like in the early months of infancy after birth, gradually these horizontal cells of Cajal, they start dying out, they start terminating out, right? So they undergo natural cell death, and that's called apoptosis. So this point also you remember that Cajal cells are not present throughout life, but of course, because as I have mentioned, they will be seen, few cells they will present even in adult life also. Okay, so now talking about the slides. So molecular cells you've seen is that I've told you, and then will be the outer granular layer. They will have more small granulocyte, but remember they will be appearing as spherocytes, because the stellate thing will not be very appreciable, because the small granulocytes. Pyramidal cells here, outer is small pyramidal, and here you'll find is small pyramidal cells. Herein you'll find you have medium size, here also medium size, and this one inner pyramidal layer has the large pyramidal cells. And granulocytes will be larger here in the inner granular layer. Don't forget that here you will find is what I have drawn here is, anybody? This is external band of Billarger, and here you have is the internal band of Billarger. Okay, so you may not be seeing it easily here. I'll show you more of slides. Yep, so identifying of a nervous tissue. So, first of all, any slide you will see, if you find such pyramidal cells, can you appreciate now? Such pyramidal cells, right? Can you see this one? Can you see this one? This one, like, so clearly they have marked it out. This one. So if you start identifying pyramidal cells, at least you will be sure of that it's a nervous tissue. Remember neurohypophysis also, not only cerebrum, cerebellum and spinal cord. There is neurohypophysis sometimes given for identification. So you just find out these pyramidal cells, there you will be able to guess. And of course the rest of them you may also find the granulocytes as well as the neuropile. Neuropile is actually the white matter, the nerve fibers in between. So that was about the okay. So now let me include one more slide so that I can describe it even more better, and let's include this slide. What do you find here? Now look here, this slide is a little more better. Why? Because here you are able to see. Now look here, you can see a lot of cells here, and they have some lacunar spaces surrounding, these cells. They have, pyramidal cells. There are a lot of pyramidal cells, they're small. Actually, they might have taken it from the upper layer. So the pyramidal cells are not very large in this view. And you also find granulocytes, and all of them, they have spaces surrounding. This also will have, help to identify the neuronal tissues. So granulocytes, the pyramidal cells, you find is, and there will be blood capillaries, the blood plexuses, remember, blood vessels, blood capillaries also, and of course the glial tissue. And here you see in the peripheral portion, because less of cellularity, less of nucleus, more of fibers. This is the plexiform layer, the molecular layer, the outer layer, and you can imagine that this is the first layer, second layer, third layer. That's how you can continue downwards. You find is the larger cells in the lower space, lower tissue. Okay, the lower layers will have large cells, and sometimes you find blood capillaries also. There, it's how you find is cerebral cortex. Okay, and let me include some more slides for better understanding. Okay, so here you are seeing is another slide. Now what you find here is this one. Okay, so now here you're seeing is they have actually, that's what I was thinking, to count down the layers. So they have done this job. You can see here that the outer surface they have mentioned is pia mater. So this is the connective tissue or the pia mater surrounding on outside. This also made up of, simple common cells on outside, a thin layer, and this is pia mater. And then you find is the outer molecular layer, this one, number 1. Can you see now? Compare. So molecular layer is having less of cellularity, more of fibrous content. The number 1 you can see is small little granule cells, right? Number 3 you find is a little larger cells. They will be pyramidal cells. And third layer again you will have granulocytes. Third layer also you have granulocytes, right? All these granulocytes, and in between you have, so you see that there is like, these are planes for the nerve fibers. Number 4 is, the inner granular cell layer, right? Fifth one is having the largest pyramidal cells, right? And the sixth one is the pleomorphic or the polymorphic layer, and the last one will be the white matter down below. Still, your purpose is not to identify the layers, right? Your purpose is to identify the slide, if it's of cerebral cortex or not, or first of all it's a nervous tissue. Nervous tissue ke liye just learn to identify the neurons like triangular pyramidal cells and the space surrounding, and to identify that it's a cerebral cortex, you'll have graded cellularity, and the cells will keep increasing towards the inner, cell size will keep increasing to the inner layers. So that's how you can find, and the outer surface will be covered by pia mater. So that's how you can see, because sometimes nowadays images are being given in your PG entrance exams. So next is, you can also see this slide. Now focus on this one. They have better given different shown here. So now this also is a nervous tissue, right? This slide is a better slide. Why? Because you are seeing again the triangular cells. The triangular cells will be the neurons, right? And of course there will be glial cells all the way. They will fill with different types of glial cells. You find also is blood capillaries, surrounding the space, and then the neuropil is actually the spaces, the fibrous space between different cells made up of white fibers. The axons running within the gray matter, they are actually neuropil, right? So you will find them also here. And because for the undergraduate exams, you are asked in your exams to identify the slides. So this one I have brought in the slide as what you see in your microscope. This slide is taken from the microscope itself, right? So the real slide taken from a microscope, so you can see how it appears in your microscope. Here one thing will be an outer surface. This, right, you will find that it will be a thin layer of fibrous layer, that's the pia matter. And can you see, you may not be able to easily identify the layers, but you can see that there are cells embedded, there are cells with lacunar spaces surrounding, right? You can see there are cells, lacunar spaces, and can you see there are fibrous pattern vertically as well as horizontal patterns. So those are the fibers, and down below it's the axons, the white matter. So, after seeing this, you can easily actually compare with the cerebellum. There you will have a clear-cut demarcation of the three different layers, which is very easy to identify. But cerebrum, at least you can identify it's a nervous tissue with the help of those cells, triangular cells in the spaces, and the graded increasing size of the neurons. Of course, you will have blood capillaries also in there. So, I think now it is complete and it is done. Okay, so there I have completed my lecture. Once again, for those who want to take screenshots, they can take the screenshots, right? They can take the screenshots. These are the Neuroglia of the gray matter. This is about the Plexiform layer, about different cells, right? Martinotti and the Fusiform cells, you can see them here. Gliomorphic Fusiform layer. And this was about the large pyramidal cells of Betz. Okay. Then this was the inner granular layer, the granulocytes, and this was outer pyramidal layer, and this was outer granular layer. This was about the interstitial cells of Cajal. These were the different cells found in the GIT, the inner circular layer, mesenchymal cells. And these were the horizontal cells of Cajal, also called Cajal Retzius cells. Remember, I taught you detail about this. They secrete reelin for the repositioning of the neurons. And this is the slide about the histology of cerebrum. Okay, you want to take the screenshots, you can take the screenshots. And okay, and now about the heterotypic and homotypic cortex between cerebrum and cerebellum, the differences, right? And the neocortex and the paleocortex, or the archaeocortex, archaeo or archipalium. This one is allocortex and this is isocortex. So there it is done. Thank you guys.